Hello friends, in this video we'll be talking about SEM, that's scanning electron microscopy. Basically we'll be discussing its principle here, not the instrumentation. So let's get started. We have some key points here. First of all, this technology was invented by Manfred von Erdőny. It's a surface microscopy rather than the microscopy we have seen in TEM, which use the electrons which go through the specimen, but here the electrons which gets reflected from the surface are used in this technology. Another thing is that it's the behavior of electrons with which the image is formed, and the most important one is the 3D image. That means the image generation is three-dimensional because it uses surface reflected electrons to create images. Now we will see the principle of SEM. It works on the principle of scattering of electrons on the surface of sample. When the electrons gets bombarded from the source, either they get reflected or they get absorbed or they get through the sample. So all the three cases are there, but in this case, in the scanning electron microscopy, the image is scanned here. That means the electrons which gets reflected from the sample are detected here and they are used in this technology. Now you can see in this diagram the electron source here. The electrons are bombarded towards the sample. Some electrons get through the sample, what we call as unscattered electrons we have seen in the TEM, and these unscattered electrons are used in transmission electron microscopy. But here we use these kind of electrons, what we call as secondary electrons, which gets reflected from the sample. So here in this case we use these electrons. In detail we can see here the electrons gets reflected back in the form of secondary electrons, in the form of backscattered electrons, in the form of X-rays, or also some Auger electrons, but only the secondary electrons are detected by the detector which are used in SEM technology, that's scanning electron microscopy. In SEM technology, electron beam scans object in raster scan pattern, that's the object is scanned side by side. That's why here the 3D image is formed. Secondary electrons are detected by electron detector, which is a part of this microscopy. Then there is a processing of these secondary electrons and eventually we get the 3D image. So this was all about SEM. Thanks for watching this video, and do share this video and do not forget to subscribe this channel.